When discussing infectious disease, it's best to start orienting where we actually are in the body. For our example, we'll take a human, and let's go and talk about a skin infection. There's a lot of bacteria there, especially a lot of gram-positive bacteria. So there's going to be gram-positive bacteria. We're showing them as spherical, and they've got this thick cell wall, and that allows the bacteria to live very well on the skin. We want to get a little bit of a closer look of the cell wall. So here's a cutout. And so the very first layer, we have the lipid bilayer. The lipid bilayer is very similar to our lipid bilayer, and that's the internal structure of the cell. On the outside, we have got that thick peptidoglycan layer, and that thick peptidoglycan layer provides the bacteria with its rigidity and again allows the bacteria to live in environments that have low amounts of moisture. Also found within the lipid bilayer are some proteins. Some of them we call membrane proteins. There might be transport channels, and those are going to have a lot of physiological and biochemical functions that we won't focus on today. We do want to mention about the lipoic acid and the teichoic acid. So they are going to be these vertical structures. The lipoteichoic acid has a lipid residue that's embedded in the lipid bilayer, whereas the teichoic acid is going to be anchored in the peptidoglycan layer. They serve two purposes. One of them is to provide a vertical structuring or scaffolding for the matrix of the peptidoglycan layers. Also, the very tips of the lipoic acid and teichoic acid will convert the bacteria to weakly adhere to different charged surfaces, such as our cell. Leads us to this last structure that is an adhesin. So adhesins is a big class of family of extracellular proteins that are going to confer the bacteria allowed to attach. Some of them are very specific for their binding specificity, so that's why we have it represented as a very complex shape. So these adhesins have very specific binding specificity. In the case of Staph epidermidis, which is a commensal on our skin, it's able to bind to healthy skin tissue. As compared to Staph aureus, it's got a fibronectin binding adhesin that can only bind to receptors that are present on diseased or inflamed skin. Whenever the bacteria is able to adhere, that's going to be serving as a trigger for the bacteria to start its replication. Once the bacteria initially adheres, it will also express more adhesins, and it's going to start binding really tightly to the receptor surface. That will also serve as a signal for the bacteria to start replicating. And each bacteria that replicates will also have a binding capability. So we're looking at binary fission, so the bacteria is able to copy of itself. So 2 bacteria become 4 bacteria, 4 bacteria become 8 bacteria. And the bacteria will start going to a critical number, and that's what we call a quorum. And that will serve as a signal for the bacteria to start releasing these extracellular components to build up its colony. Once the bacteria reach that critical number, they start secreting these extracellular components, including extracellular DNA, amyloids made up of peptides, and this extracellular polymeric saccharide. And all of these components are very sticky, allow the bacteria to stick to themselves and more tightly adhere to that receptor surface. Let's take a big picture. Here we have the bacteria that's initially attaching, it's replicating, it's reaching that critical number. And as a net result, we're going to be having this extracellular large biofilm. So the biofilm is considered like a macrocolony, and this mature structure is going to protect the bacteria from immunological insults, and it will also protect the bacteria from antibiotic from penetrating. Whenever the bacteria get to a critical number, it's going to serve as a signal for the bacteria to start releasing some degradation enzymes. These enzymes are only released at the top layer of the biofilm, which was going to release free bacteria to infect other surfaces. However, with the mature biofilm, the base of it has irreversibly attached and is going to stay persistently colonized. In the case of commensals with healthy tissue, these biofilms are actually beneficial for the bacteria to be present on our skin for a long duration. However, if you have a scenario and your epidermis layer is cut and the bacteria gain access to deeper tissue, that's going to be a problem. So our commensals that are not particularly virulent, they cannot penetrate to deeper tissue. However, some bacteria, they have evolved a defense to get past our extracellular matrix. So here we're showing the hyaluronic acid, which is present with our epithelial cells, and the collagen layer that is a part of our extracellular matrix of our skin. So the bacteria produce enzymes, so it's that the collagenases are going to be breaking down the collagen, and the hyaluronidases are going to be breaking down the hyaluronic acid. And both of these enzymes are going to confer the bacteria to get into subdermal layers where they'll start interacting with our immune cells. If we have a break in the epidermal layer, one of the first immune cells that will be encountered are macrophages. Macrophage will be able to phagocytose, recognize bacteria, and they'll start producing chemotactic factors and cytokines, which will then recruit our neutrophils to migrate from our blood vessels to the tissue. So we have this other immune defense called complement. So this is represented as this circle. Complement C3 is circulating around all the time. And what happens whenever you get a cut in the skin, the complement C3 will spontaneously break apart. Some bacteria have developed a cloaking defense against this tag and recognition system, and it's called a capsule. What happens, the C3b will still get deposited. However, the sugar capsule is going to serve as a barrier to prevent the receptor that is found on the phagocyte from actually physically interacting with the C3b. If you have commensal bacteria that are not encapsulated, they will be completely cleared and destroyed by our macrophages and neutrophils. However, in encapsulated bacteria that have enzymes that can break through our extracellular matrix, they can penetrate deeper and get into the bloodstream. Additionally, some bacteria have another virulence factor, an exotoxin that can destroy our cells. Coming back to the scenario of a local infection from an exogenous strict pathogen, the bacteria will attach, colonize the skin tissue, and start secreting the exotoxin, which is highly soluble. The toxin can travel through the bloodstream and lymphatics to the lymph nodes. The superantigen exotoxin works by binding to and locking the interaction of the T cell receptor and the MHC on an antigen-presenting cell. This causes activation of the T helper cell and macrophage, represented by arrows up, leading to massive cytokine production and shock. In this case, the bacteria remain local, but the superantigen causes systemic shock,